Water and oil and water, W/O/W emulsions are a clever way to encapsulate proteins and enzymes, protecting these delicate biomolecules and controlling their release. In this system, tiny droplets of water containing the protein or enzyme are first encapsulated within an oil phase, which is then dispersed into a continuous outer water phase. This layered structure creates an extra barrier that shields the proteins from harsh external conditions such as changes in pH or the presence of degrading agents. Imagine you're trying to protect a fragile artifact during a long journey. This emulsion acts like multiple layers of protective packaging, ensuring that the protein remains stable until it reaches its destination. How does this benefit practical applications? For one, it enhances the shelf life of therapeutic proteins and enzymes used in industrial processes, making them more robust and effective over time. One exciting application of W/O/W emulsions is in the field of drug delivery. Proteins that act as drugs can be encapsulated in these emulsions, allowing for controlled release once they reach a specific target in the body. For example, an enzyme that breaks down a disease-causing molecule could be delivered directly to a site of inflammation, ensuring that it is released gradually and in a controlled manner. Isn't it fascinating how such a tiny droplet can perform such a critical task? The answer lies in the emulsion's ability to finely tune release rates by adjusting the thickness and composition of the oil layer. In the realm of food science, these emulsions have also found a niche by protecting sensitive enzymes that contribute to flavor development or nutritional enhancement. By encapsulating these enzymes, food manufacturers can ensure that the enzymes remain active during processing and storage, only becoming active when needed, such as during cooking or digestion. Could this technology change the way we think about processed foods? Absolutely, because it allows for a more dynamic interaction between ingredients, potentially leading to products with improved taste and nutritional profiles. Another intriguing use is in biotechnology research, where W/O/W emulsions serve as microreactors for biochemical reactions. Researchers can encapsulate enzymes in these droplets to carry out reactions in a highly controlled microenvironment. This method not only minimizes waste, but also speeds up reaction times by concentrating reactants within a confined space. How does this benefit scientific research? The answer is that it enables the study of reaction kinetics and enzyme behavior in conditions that closely mimic those in living organisms, providing valuable insights into enzyme efficiency and stability. Furthermore, these emulsions are finding applications in cosmetics, where sensitive bioactive compounds need to be delivered to the skin in a controlled manner. By encapsulating proteins and enzymes in a W/O/W structure, cosmetic products can offer extended release of active ingredients, enhancing their performance while reducing potential irritation. Have you ever wondered how your favorite skincare product can deliver benefits over several hours? The answer might be hidden in the sophisticated microencapsulation techniques used during its formulation. In summary, water and oil in water emulsions for protein and enzyme encapsulation showcase how innovative approaches in microencapsulation can protect delicate biomolecules, extend their activity, and enable controlled release in a variety of applications. From drug delivery and food processing to biotechnology and cosmetics, this multi-layered system not only safeguards sensitive components, but also opens up exciting opportunities for developing products that are both effective and reliable, all by harnessing the power of tiny droplets engineered to perfection. Water in oil and water. Emulsions consist of aqueous droplets dispersed within oil, which is then surrounded by another aqueous phase. This double emulsion system allows for the encapsulation of hydrophilic molecules, such as proteins and enzymes, while protecting them from degradation. The structural integrity of these emulsions depends on interfacial tension, emulsifiers, and processing conditions that ensure stability over time. Researchers have used W/O/W emulsions to encapsulate enzymes such as lipases and proteases, preventing their denaturation while ensuring controlled release in industrial and pharmaceutical applications. This system functions like a soft-boiled egg, where the inner core remains liquid, protected by an oil layer. egg white, and an outer aqueous layer, shell membrane, preventing premature exposure to external conditions. Microfluidics provides superior control over the formation of W/O/W emulsions, ensuring uniform droplet size and stability. Devices using flow focusing and co-flow geometries allow the controlled encapsulation of proteins and enzymes, reducing the risk of aggregation or inactivation. The precise control of flow rates and surfactant concentrations optimizes droplet formation and enhances encapsulation efficiency. A microfluidic chip with a double emulsion generator was used to encapsulate lactase enzymes, maintaining their bioactivity for extended release in lactose-free dairy products. This is similar to using a high-precision syringe to fill capsules with medicine, ensuring each dose is exactly the same and effectively sealed for long-term use. To maintain stability, different surfactants are needed at each interface. Hydrophilic emulsifiers stabilize the outer water phase, while lipophilic ones stabilize the oil-water interface. The choice of surfactants affects droplet coalescence, permeability, and retention of bioactivity. Optimized emulsifier combinations allow for prolonged enzyme protection and controlled diffusion rates. Using polyglycerol polyricinoate for the oil phase and polysorbate 80 for the aqueous phase enhances the stability of W/O/W emulsions, encapsulating lysozyme, preventing premature leakage. This is like using a double-sealed thermos, where the inner and outer layers are specifically designed to prevent heat loss and maintain the temperature of the liquid inside. The release kinetics of encapsulated biomolecules depend on factors such as droplet permeability, external triggers, temperature, and osmotic pressure. Enzyme-loaded W/O/W emulsions can be engineered for slow diffusion through the oil phase or rapid burst release upon external stimuli. This controlled release ensures that proteins and enzymes remain active at the desired location and time. W/O/W emulsions with pH-insensitive polymers in the oil phase have been used to encapsulate pepsin, releasing it only under acidic gastric conditions for targeted digestion. This functions like a self-dissolving pill capsule, protecting its contents until it reaches the correct part of the digestive system, where it releases the active ingredients. Enzymes are highly sensitive to environmental conditions such as temperature, shear stress, and organic solvents, which can lead to loss of function. Y/O/W emulsions act as a protective barrier, shielding encapsulated enzymes from denaturation while preserving their catalytic activity. The controlled microenvironment within the droplets provides stability in industrial and pharmaceutical applications. Encapsulating amylase in W/O/W emulsions has been shown to protect it from high temperatures during food processing while ensuring sustained enzymatic activity. This is like keeping fragile biological samples in a sealed, temperature-controlled container to prevent spoilage or degradation before use. In medicine, enzyme-loaded W/O/W emulsions provide a strategy for enzyme replacement therapy, ensuring controlled release in targeted tissues. By tuning the emulsion structure, drug carriers can enhance enzyme bioavailability, improve absorption, and minimize immune response. The oil layer serves as a diffusion barrier, regulating enzyme exposure and extending therapeutic effects. Encapsulation of pancreatic enzymes in W/O/W emulsions allows gradual release in the intestines, improving digestion in patients with pancreatic insufficiency. This works like time-delete medication that dissolves gradually, delivering its effects over an extended period rather than all at once. W/O/W emulsions are increasingly used in food engineering to encapsulate bioactive compounds such as probiotics, vitamins, and functional proteins. The dual-layer structure enhances stability, prevents degradation from gastric acid, and allows targeted release in the intestine. Encapsulating enzymes in food emulsions improves processing efficiency and enhances digestion upon consumption. YN/O/W emulsions containing bromelain, a pineapple enzyme, have been developed for meat tenderization, allowing controlled enzyme activity for better texture and flavor. This is similar to using a marinade that slowly releases flavors over time, ensuring deep penetration without overwhelming the final taste. Encapsulated enzymes in Wu/O/W emulsions serve as catalysts for industrial processes such as biofuel production, textile processing, and waste degradation. The emulsion system ensures enzyme protection in non-aqueous environments while enabling controlled reaction rates. By optimizing oil phase composition, researchers can enhance enzyme solubility and reduce inhibitory effects. Wu/O/W emulsions containing cellulase enzymes have been used for bioethanol production, improving cellulose breakdown in lignocellulosic biomass. This is like embedding cleaning agents in a protective gel capsule that slowly dissolves, ensuring sustained release and prolonged action over time. Despite advances, challenges remain in achieving long-term stability, optimizing large-scale production, and preventing enzyme leakage. New approaches, such as polymer-stabilized emulsions and microfluidic-assisted fabrication, are being developed to overcome these limitations. Future research will focus on integrating stimuli-responsive materials that trigger enzyme release upon specific environmental cues. Recent studies have demonstrated temperature-sensitive W/O/W emulsions that release enzymes upon exposure to body heat, improving transdermal enzyme delivery for medical applications. This is like smart textiles that adjust insulation based on body temperature, providing adaptive protection in changing conditions.